I find aging to be one of the most pressing and also most fascinating problems of biomedical research these days. Everybody ages, and our chance of developing disease increases proportionally as we age. So, aging is the cause of all major diseases. And I was always thinking that, well, if we find a way to slow down aging, then we'll prevent all the diseases. So it is preventative medicine. My name is Vera Gorbunova. I'm professor of biology and medicine and the co-director of Rochester Aging Research Center. We study aging, longevity, and cancer. Aging field is changing all the time. This is one of the very rapidly developing areas of science. So many things change pretty much all the time. But a lot of progress happened. And first with model organisms, people were able to extend lifespan of fruit flies, of worms, and now of mice. We moved to naked mole rats. This is even more fascinating creature. It's a very tiny rodent, the same size as a mouse, but they live more than 10 times longer than mice. 40 years was documented lifespan for naked mole rats. And they are resistant to cancer, they are resistant to neurodegenerative disease, heart disease, osteoarthritis. So this is a real model of healthy aging. We started culturing cells from naked mole rats, and then we noticed that they secrete very large quantities of the molecule called hyaluronic acid. And we were very excited, and then we started investigating how this molecule affects the cells, and then we find out it slows down progression of premalignant cells. We genetically engineered the mouse to make more hyaluronic acid by putting a gene from naked mole rat into the mouse. And now these mice get fewer tumors. They're somewhat protected, not to the same extent as naked mole rats, but they get, let's say, 50% less cancer. If we could reduce cancer incidence in people by 50%, wouldn't that be amazing? And now we are working on developing small molecules that can achieve the same result by targeting the same genes within human cells. So here you can see an example. We start from naked mole rat and we go all the way to developing pharmaceuticals that can be used in humans. We were looking at other factors that may be different in long-lived species versus short-lived species, and we tested several candidates, and we found that the ability to repair breaks in DNA correlates very strongly with lifespan. Long-lived species are very good at repairing DNA breaks. And then we identified a specific gene that was responsible, and the product of this gene called sirtuin-6 was a more active enzyme in long-lived species. We hope that by activating SIRT6, so finding a way to regulate it, we would be able to reverse some of the processes that happen with age, so I would call it rejuvenation. Sirtuin-6 controls epigenetic sort of chromatin structure. So this is the way how our DNA is packaged within the nucleus. Everything needs to be organized. DNA is a very long strand that needs to be wrapped in a certain way. But as we get older, this organization unravels and becomes disorganized, and it makes it more difficult for the cell to function properly. What sirtuin-6 its job is to help and put DNA, wrap it back together closer to its youthful conformation. So what we hope to achieve is to have a way to stimulate SIRT6 in people, past certain age. You would take this pill or whatever intervention it ends up being and rejuvenate your chromatin. Right now we are doing a lot of omics studies where we study the transcriptomes, proteomes, metabolomics across multiple species, and we look for factors that are linked to longevity. So using our genomics facility, proteomics facility, metabolomics has been extremely valuable, having all these resources right here. And that was really critical for our discoveries. The University of Rochester was very supportive from the beginning to these unconventional research approaches. the environment is so collaborative. We have both evolutionary biologists, molecular biologists, and physician scientists. So this is kind of the environment we need because we interact with all of these different fields. I hope that my work will contribute to this future of human civilization where we can all live our lives to the fullest and just remain healthy for as long as possible.